Welcome back. We're continuing on with our antibody discussion. Antibodies do not destroy antigens. They inactivate and tag them when they form antigen-antibody or immune complexes. Antibodies use neutralization, agglutination, precipitation, and complement fixation as defensive mechanisms. Beginning with neutralization. It is the simplest, but one of the most effective defensive mechanisms antibodies have. Antibodies block specific sites on viruses or bacterial endotoxins, preventing the antigens from binding to receptors on tissue cells. The immune complexes then undergo phagocytosis. We've heard the term agglutination when we studied blood typing. Antibodies can bind the same determinant of two different antigens at the same time. Each antibody has two arms, with a variable region capable of binding to one antigen. This allows immune complexes to become cross-linked into large lattice-like clumps referred to as agglutination. We can see this up here in this box. Precipitation is when soluble molecules instead of cells, are cross-linked into complexes. You can see them here. The complexes precipitate out of solution, making them easier for phagocytes to engulf. Lastly, antibodies use complement fixation and activation as a defensive mechanism. This is antibodies' main defense against cellular antigens like bacteria and mismatched red blood cells. When several antibodies are bound close together on the same antigen, the complement binding sites on their stem regions are aligned. This alignment triggers complement fixation, which leads to cell lysis and other complement functions, like amplification of immune response or opsonization. You can see this here. Figure 21.15 shows the mechanisms of antibodies. Antibodies attach to antigens, creating antigen-antibody complexes. This immune complex inactivates antigens by This immune complex inactivates antigens by neutralization, agglutination, precipitation, and these three methods enhance phagocytosis. The immune complex fixes and activates by the complement inserting MACs and causing cell lysis. Parasitic worm infections require a different immune attack strategy. Worms are too big for the regular PLAN plan attack. P standing for precipitation, L for lysis by complement, A for agglutination, and N for neutralization. So, IgE antibodies mark the surface of the worm for destruction by the eosinophils. Eosinophils bind to the exposed stems of IgE, which triggers eosinophils to release their toxic contents onto the prey, lysing it from the outside. Monoclonal antibodies are commercially prepared pure antibodies for a specific single antigenic determinant. They are produced by hybridomas, cell hybrids formed from the fusion of tumor cells and a B cell. The tumor cell portion allows cells to proliferate indefinitely, while the B cell portion allows production of a single type of antibody. These monoclonal antibodies are used in research, clinical testing, and cancer treatment. In summary, antigen-antibody complexes do not destroy antigens, but instead prepare them for destruction by our innate defenses. Antibodies go after extracellular pathogens and do not invade solid tissue unless a lesion is present. One recent exception was found. Antibodies can act intracellularly if they are attached to a virus before the virus enters the cell. Antibodies activate mechanisms that destroy the virus.